Thank you very much, Paul, for that kind introduction. And I feel good right now. I think it was good I'm doing an early morning talk, because maybe by the afternoon I won't feel so great. So, and thank you to Tim for the introduction, because it just made me realize how much is behind what I'm saying that I just forget about. So really, just interrupt me if you have any questions, if anything is unclear, because, as I say, I think I tend to assume a level. Oh, am I not moving this forward?
I wasn't able to get that.
You weren't able to get it to work? Okay, all right. It's not me then. Okay, all right. Yeah, so I'm going to give you a history of Alzheimer's disease genetics. I'm not going to go back to the very beginning, but as Paul mentioned, I have been working in the field for 30 years, so you're going to get 30 years in an hour or less. So this is what I'm going to try and cover. So we're going to talk first, in a few slides, about the Mendelian causes of Alzheimer's disease. So what Tim talked about, we'll set you up for that. The second bit will be a little bit about ApoE, which is sometimes called a codominant effect. And then we will move on to talking about genome-wide association studies, since that's what everyone's going to run in the next few days. So common low effect variants in the genome. And I will talk also some about how to go from those, the results of a GWAS, to actually really trying to understand what underlies those signals. And lastly, we'll talk about whole genome and whole exome sequencing and identifying rare variants. So a lot to cover. So you'll see this slide lots of times, and it comes from a review by Terry Manolio, now from 2009. But I think it's a good slide to illustrate how we think of the genetic architecture of pretty much any disease trait, for sure. In that you have, for many diseases, you have this small group of individuals who may carry very rare alleles that cause Mendelian disease. And so on this graph, you have allele frequency on the x-axis going from very rare to common. And on the y-axis, you have effect size, so low effects, sort of 1.1-fold, up to very high, and obviously fully. So these ones here are fully penetrant. If you carry the allele, you're going to get disease, right? So this is where our familial Alzheimer's disease genes lie. And then on the other side over here, at the top, maybe not really as high as that, but ApoE is pretty strong effect, as you guys know. So ApoE would be here. High effect common variants. And actually, there are not that many in the population. Macular degeneration has a similarly strong genetic risk factor, but most other disease traits do not have variants that fall in this group. Most of their variants would fall here, which are common variants that have a small effect on risk individually. And so these are the ones, this group here is, we use GWAS, or genome-wide association studies, to identify these. And then to identify low-frequency variants or rare variants with smaller effect, we use whole genome or whole exome sequencing. So this sort of group here, we're using a sequencing approach, and here we're using a GWAS approach. Okay, so I do have one pedigree, Tim.
Yes.
All right. And hopefully, just looking at that inheritance pattern, you can guess what the inheritance pattern of most cases of Alzheimer's disease, most cases of the familial version are. They're autosomal dominant, so carrying one copy of the disease allele is sufficient to cause the disease. And these are very rare in Alzheimer's disease, but they were tremendously helpful in beginning to sort of unpick the genetics. And to find the disease genes, we used linkage analysis, which Tim mentioned. And so and to do this, we were using, I would say, pretty primitive genetic maps back in 1987, in that each chromosome had a relatively small number of DNA markers that you were using to follow inheritance. And actually, back in 87, it was all done using a Southern blot, which is probably there's probably nobody in the room except me that's used a Southern blot. And actually, they were named after a man called Ed Southern. It's not a geographic point on a compass. So for Alzheimer's disease, we actually had I would say from a genetic perspective, a really big clue, which was that everybody with Down syndrome who lives long enough will get Alzheimer pathology in their brain, and the majority of them will show cognitive impairment with age. And everyone with Down syndrome is trisomic for chromosome 21, which means they have 3 copies instead of 2 copies of chromosome 21. And so that strongly suggested that there was something on chromosome 21 which could influence risk for Alzheimer's disease in a big way, in that if you carried an extra copy, you were pretty much guaranteed to get Alzheimer's disease. And in 1987, the amyloid precursor protein gene was cloned. So for those of you who don't work on Alzheimer's disease, I don't think I have a slide in here, actually. The pathology of Alzheimer's disease has amyloid plaques and tangles. And actually what happened was that several investigators, George Glenner, Colin Masters, a few others, purified the protein from the brains of people with these either Down syndrome patients or Alzheimer patients, and then sequenced the peptide that they purified, and then screened cDNA libraries to find the gene that encoded that peptide. And what they found was that the amyloid peptide, which is 40 amino acids long, wasn't actually the full length of the protein. It was a fragment of a protein that's encoded by a precursor protein, APP, or amyloid precursor protein. And APP is encoded by a gene on chromosome 21. So that all seemed pretty neat. We have the major protein in Alzheimer pathology encoded by a gene on chromosome 21, and everybody with Down syndrome having Alzheimer pathology if they live long enough. So actually, the point at which I joined John Hardy's lab was just at a time when there were 2-3 papers in Nature and Science that said, amyloid is not the gene. So I always tease John because he has his name on that paper and on this one, whereas I only have my name on that one, right? So, and the reason that everybody got it wrong is an important genetic lesson, right? In that the assumption was that all of these families that looked like they had an inheritance pattern like that had a mutation in the same gene. So that if you saw a recombination event in a family between the disease and a polymorphism in APP, then APP could not be, a mutation in APP could not be causing the disease. The problem was the assumption was wrong. As you can see, there are at least 3 genes, and so all those papers that said amyloid is not the gene were looking at families that actually carry mutations in other genes that I'll mention in a minute. They didn't actually show any linkage to chromosome 21. And so, obviously, they didn't have a mutation in APP that was causing the disease. But we were actually fortunate that we had this family, which we already knew from studies that we had published earlier actually did show linkage to chromosome 21. So that means that when you take any polymorphic DNA marker on chromosome 21, pretty much, and genotyped everybody that we had samples from in this family, all of the people with, who had disease, which are the colored-in people here, carried the same allele of a polymorphism in a whole, actually pretty much the whole region of chromosome 21. It was a pretty large region. And so we could say that in this family, there's the same region of chromosome 21 was segregating with the affected individuals in the family. So there was linkage in this family between the disease and a specific polymorphism in APP. So based on that information, we then sequenced the APP gene. Actually, back then, this was a really big task. So there are 18 exons in APP, but the amyloid sequence is actually is encoded by exons 16 and 17. So we sequenced exons 16 and 17 in everybody in this family, and we found a single base change that resulted in an amino acid change at codon 717 of a valine to isoleucine. So every affected individual in this family carried that variant. None of the unaffected individuals in this generation carried that variant. But obviously, as you can imagine, in this lower generation where people hadn't yet reached the age of onset, there were some people who carried that variant. And all of those people were the children of an affected individual. So that was good. It seemed like we had a variant that was segregating with disease. So then we screened, now seems incredibly naive, but it's what everyone did. We sequenced those exons in 100 people, right? And we didn't find it. So we said, oh, that's got to be the cause of disease. So now there are public databases. Some of you may have looked in the exact browser, which is a browser with 64,000 people in it. And I can find you some of these mutations in that database because they exist obviously at a high enough frequency that you can find them in some relatively unscreened populations. So, I mean, we'll come back to what that might mean. So that was one thing. So it wasn't present in the normal population from which this family came, based on 100 people. And then we sequenced those same 2 exons in other families that we had at St Mary's in London that had autosomal dominant form of Alzheimer's disease. And we found one other family that carried the same mutation. So now we had 2 families with the same mutation in APP and no cases in our normal population. So that was the evidence that was sent to Nature and was resulted in that paper. And over time, as you'll see in the next slide, it was clear that this actually, although this is even amongst familial Alzheimer's disease, it's not the major cause of disease. But I would say at that time, given the sparsity of markers across the genome, it was the easiest one for us to find because obviously we could go look at 21 based on Down syndrome. So the majority of autosomal dominant families in Alzheimer's disease have a mutation in this gene, presenilin 1. And presenilin 1 was found by pure reverse genetics, and it was a hard slog, right? So by that time, you can see it's now 1995 when this was published, there was a better map of the human genome. So 1987 was actually when the first genetic maps with any decent number of markers were generated. PCR came around 1990. So polymerase chain reaction is a way that allows you to get rid of that southern blot and now just do things much more quickly and in a larger number of samples. And so this was the, I think it's about 1992, the first linkage in these Alzheimer families to chromosome 14 was reported by Peter Hislop and colleagues. And it then took 3-4 years from then. So then it was clear, actually, from those linkage studies that there were a lot more families that showed segregation with chromosome 14 markers than there ever had been for chromosome 21. So that was the first indication that this was likely to be more common. And so, but even though the maps were better, we still didn't have every piece of DNA cloned, right? So, and as Tim was mentioning, so for linkage, to narrow down the region, you're using that recombination in meiosis to be able to determine which bit of the chromosome your disease gene is lying in. And these families are actually, first of all, pretty sparse, right? There are not many of them, and you can see that many people who you know were affected are dead. So you don't always have samples from them. Sometimes you do, but not always. And so they're actually not very powerful for recombination mapping because you don't have actually that many people with information. So over a period of several years and across many labs, the region was narrowed down to, I think, about 8 or 9 megabases or million base, 8 or 9 million base pairs through recombination events. So basically, you're looking for those recombination events to define where the disease must lie. And of course, as I mentioned with the APP story, you are assuming, again, that the disease is caused by one gene, right? And at least in this case, there were many more families that showed linkage to chromosome 14, so you could restrict your use of recombination events to families that also showed linkage. So to measure, the way you measure or you put a statistical value on linkage is using a LOD score, which is the log of the odds of the observed over expected and then you attach a p-value to that. And so significant, the most important thing to remember about this is that a log score of 3 is equivalent, is significant association in p rate. So there are many more families that showed a log score of 3, which allowed you to use these recombination events to narrow down the region of the chromosome. So then we had to clone that 9 megabases of DNA and work our way through that region, sequencing in families to find the gene. And yeah, I can see your head, Tim. Yeah, that was the hardest slog of any of these, because the other two really came out of as candidate genes, right? But this one, we really were using reverse genetics. You were saying, which chromosome out of the 22, well, we knew it was an autosome, so out of those autosomes, which autosome was it on? So that was the first step. We got it down to 14. Then those recombination events in families narrowed it down from the full length of chromosome 14 to a region of about 8 or 9 million base pairs. But then you actually had to clone all of that DNA and sequence it in families to find the gene. And so this was Peter Hislop's group, Robin Sherrington, who reported the first mutations in presenilin 1 back in 1995. And then actually, so this was really, I would say, a triumph of the Human Genome Project, in that when this gene was cloned, or when they found these mutations, so they were kind of sneaky, actually, in the paper. They did not put the DNA sequence. They only put the amino acid sequence. And that was because they knew from their studies that there was homology. So, but of course, because you have we know what codons code for amino acids, you could actually, with some redundancy, go backwards. And it became clear that there was a homologue of presenilin 1 on a different chromosome. And Jerry Schellenberg and Alan Wiseman and the people at UW had a very big family, or set of families, called the Volga German families, which all carried a mutation in presenilin 2. And so you can see this was 1995. I think this was June 95. And in September 95, Peter Hislop's group and the UW group found a mutation in that Volga German family in presenilin 2. And it was really done so quickly. Well, the UW group knew there was linkage to chromosome 1, right? Because they had done the segregation analysis in those families. And then when they saw that the homologue of presenilin 1 mapped to chromosome 1, that was target number 1. That was where you needed to go. So then sequencing presenilin 2 in those Volga German families identified the presenilin 2 variant. And in all the years since then, I would say there's not been, so I would say the vast majority of autosomal dominant AD is caused by a mutation in one of these 3 genes. That there really are no big families where we've not found a mutation. So there may be other genes, but they are not present in big families where we're actually able to do have been able to do studies. I mean, now with a lot more sequencing going on, we may turn up some other genes, but these account for the vast majority of the autosomal dominant forms of Alzheimer's disease. In 2009, the Indiana group reported a family where the inheritance is recessive, and this is a mutation also in APP. But these are, I would say, this is the only family, I think, that's been reported in the literature. All of the others are dominant families. So now that those genes are being cloned for about 20 years, we have a pretty good idea about what proportion of these autosomal dominant families are caused by mutations in different genes. And so what you can see here is that by far the most common gene to be mutated is presenilin 1. So whether you do it by the number of families or the number of mutations, around 80% of these families or mutations are in presenilin 1. And then about another 15% are in APP, and about 5% are in presenilin 2. And as you can see, so if we take as an example the presenilin 1, so in the literature there are around 400 families that have been reported, and you can see there are almost half as many mutations, 177 mutations. So that means most families who come to a clinic have a novel mutation. Right? So this is actually pretty challenging, because if you find a mutation that's been reported before in an Alzheimer pedigree, you can say this mutation is likely to be causative for the disease. But when you find a novel mutation, you don't know whether it's a benign polymorphism or whether it's a pathogenic mutation causing disease. Because the way these arise is because somebody comes with a family history and they go to a clinical geneticist or a neurologist and they have their genes sequenced in one person. So you're never looking at segregation in these studies. So that is why I mentioned that exact database, because one of the things you can do is go to a big population database and say, can I find this variant in there? And if it's not in there, and you have it in one of these families, then it's very likely to be causing disease. But what happens when you find it in there as well? Then are there some familial Alzheimer's disease families in this exact database of, they're not random individuals, right? This is sequencing from a lot of different NIH projects. And one of the cohorts is from Finland, which actually does contain some Alzheimer cases. So it is still possible that it's causing disease, but it's kind of blurring the lines and it's harder to interpret. And this is the other reason I think it's harder to interpret. So all of those early families in the 90s that people were studying had an early onset of Alzheimer's disease, as well as having that autosomal dominant pattern of inheritance. So some of the families have an onset in the 20s, right? The majority of them are almost all of them are before 60, and probably the most common age range is in the 40s and early 50s. So actually it's much easier to see, they all look completely penetrant. In those families, if somebody carries a mutation and then they will get disease by within the decade of the onset of other family members in their families. And Jason and I are part of a study called the Dominantly Inherited Alzheimer Network, and we've been doing an observational study in about 450 people from families with these mutations. Among all those families and all those people, I think there's one person I can think of who is 10 years beyond the age of onset in his family, and he's still cognitively normal. So it's extremely rare. Or at least based on those data sets, it's rare, right? So this is some data. I'm not going to get anywhere near through the slides, but we can stop when we want to. So this was back in 2012. So with Richard Mayer, I and Tatiana Farud have been collecting, and a bunch of other sites actually. I mean, have been collecting late onset AD families through this study called NIA LOAD, or late onset Alzheimer's disease. So these families all have an age of onset above 60, or a mean age of onset above 60. In some families, there are a few people with onset in the late 50s. And we collected 1,000 families. And among those 1,000 families, Carlos and I identified there were 439 families where there was a family history of dementia in at least 4 people in the family. So these were the most consistent with idea of having some autosomal dominant disease with age-dependent penetrance. And then we sequenced, yes.
First-degree relatives?
Yes, they were first-degree relatives. Everyone was a first-degree relative. Thanks. And so by this time, we knew, obviously, about APP and Presenilin 1 and Presenilin 2. But we also knew that frontotemporal dementia could be caused by mutations in the tau protein, or MAPT, granulin, and C9orf72. And in these families, you didn't have to have autopsy to be enrolled in this NIA LOAD study. So only about a quarter of the families were autopsy-proven Alzheimer's disease. And even then, it's in one person in the family. And Alzheimer's disease is really common in old people. So we sequenced these 6 genes in one person per family, and to look to see whether we could find mutations. So we found 2 different mutations in Presenilin 1, and these were mutations that had been reported in those early onset autosomal dominant families. So we knew they were pathogenic in those other families. And here, sometimes they segregated with disease, sometimes they didn't. It was but it was 1.6% of these late onset families carried a known mutation in Presenilin 1. And even worse, you can see there were just over 1% had an expansion of the repeat in C9orf72, which is associated with frontotemporal dementia and ALS. So about 1% of these sample families that we thought had Alzheimer's disease probably have frontotemporal dementia. And similarly, 0.7%, so almost 2% of these families carried mutations in genes that cause frontotemporal dementia, not Alzheimer's disease. So these were all collected in ADRCs, or centers where people are expert at diagnosing Alzheimer's disease. But in the absence of an autopsy, so clinically they look like Alzheimer's disease, but genetically 2% of them were frontotemporal dementia. Right? And a further 1.6% carry mutations in these autosomal dominant early onset genes. So it's one of my pet peeves when people call these genes early onset genes. These genes, that's an ascertainment bias big time, right? If you sequence early onset families, you will find mutations that you can say are early onset genes, but it's because you sequenced early onset families. There are numerically many more families with mutations, at least in Presenilin 1, with late onset Alzheimer's disease than there are with early onset Alzheimer's disease, because early onset is so rare, right? So this is 2% of these big families with late onset Alzheimer's disease have a Presenilin 1 mutation. And one of these is A79V, and I know Tom Bird at University of Washington also has a late onset AD family with that. And we found that mutation in early onset AD, late onset familial AD, and late onset sporadic AD. That is a highly modifiable penetrance of that variant. Yes?
So is that a loss of function? Do we know?
No, I mean, there's a big argument, which I'm not going to go into, in the field about whether these mutations are loss of function or gain of function. Or, yeah, so I mean, at a molecular level, if you look at A beta levels, it doesn't look that dissimilar from other presenilin mutations. So, I mean, that would argue that whatever the modifier is, it's kind of beyond the processing of APP. Yes?
So in the clinic, we typically see a family of late onset where we see perhaps an autosomal dominant pattern.
Mm-hmm.
And we typically won't.
Yeah, right. Yes.
Because they're late onset.
Yes.
And I just wanted to hear your thoughts.
Yeah.
Is that
I think if there's a really strong family history, I would do it, right? Especially if you know that they don't have many E4 alleles. I mean, if everyone's homozygous for E4, it's probably that. But in the absence of that, I think it's worth looking at this. I mean, my impression from talking to people who order these tests, they're much more likely to order a test like this in a sporadic early onset case than they are in a familial late onset case. And I think that's probably the wrong strategy. I mean, it's now been shown that there are de novo mutations that lead to sporadic early onset AD in a proportion of them, but it's pretty rare. And so all of the genetic testing pretty much in North America is done by Athena Diagnostics, and they're not very keen to divulge what proportion of the people that get sequenced actually carry these mutations, because it would be kind of interesting to know, right? And we know from our Diane study that when we apply very strict criteria, we find a mutation in at least 50% of people. But I'm sure I know the Athena Diagnostics values are much closer to 1%. So I think if you focus on family history and having a pretty narrow range in age of onset, you're much more likely to come up with a family that carries a mutation than if you just go for early onset with no family history. Yeah.
I had a woman last year who was diagnosed at age 50. Both her parents made it into their 80s without Alzheimer's before dying. And her ApoE was 2.2. So I sequenced her and she had an 879B.
Oh, she did. Yeah. That mutation I don't know what the population frequency is, but that is one of the ones that's in exact database, right? And it really has a very variable penetrance from family to family. So it is hard from a genetic testing point of view to actually advise people when you find it about what it means for their risk for disease. So, yes?
So last year we learned that there are relatively few pure cases of Alzheimer's. There's a mixed Lewy body disease,
right,
all mixed in. What in your families were they all considered pure Alzheimer's?
No. That very first mutation, one of the reviewers in Nature in 1991 said, this isn't Alzheimer's disease, they got cortical Lewy bodies. So, and very interestingly now in the Diane study, Nigel Kent, who's the neuropathology core leader, we now have 24 brains, I think it is, from these Diane families. And of all the different pathologies that you get in late onset Alzheimer's disease, most of these early onset AD families don't have any of those other pathologies. The only one they do have is Lewy bodies. And it's about, I think it's 60% of the people who've come to autopsy have plaques, tangles, and Lewy bodies, cortical Lewy bodies. Yes?
Do people in Diane have cerebrovascular disease, white matter disease, or strokes?
No. With the exception of people who have mutations that are associated with duplication of APP or so the APP mutation people, some of those people have more amyloid, so the APP duplication, so like Down syndrome, right? You see, you do have this vascular component, and if you have a duplication of APP, you can have that vascular component and you can have strokes. But the other families, there is no vascular disease, no white matter disease there. So it really sort of separates them out, and I think it's really interesting that such a high percentage of them have cortical Lewy bodies, because that really links Lewy body pathology to Alzheimer's disease mutations in a very different way to these other pathologies. So TDP43 pathology, vascular pathology, are other pathologies that you find in really old onset AD cases very, very frequently. Right? Most AD in people over the age of 75 is not pure plaques and tangles. They've got these other pathologies. But in these families with the early onset form of disease, you don't find any of that other pathology except the Lewy bodies.
Alison, you mentioned implications of APP cause vascular strokes. Like, is it hemorrhagic due to amyloid angiopathy or ischemic?
Yes. Hemorrhagic. Yes. Yeah. Yeah. Yes.
10 or so minutes, 15 maybe.
Yes. Okay. So we're going to move a bit faster. All right. So okay. I don't know who said that, right? So APOE. Probably everybody knows about APOE, so I will do this bit pretty fast. So actually, back in 1993, Alan Rose's group was the first group, and they actually reported APOE through linkage. They did linkage in late-onset AD families, found linkage over the region that contained the APOE locus. We knew that APOE was in an amyloid plaques. They found APOE bound to amyloid in the cerebral spinal fluid. So they looked at, and this polymorphism in APOE was already known in the literature because of its association with heart disease. So this polymorphism is, as Tim mentioned, something we refer to as alleles, but actually it's a haplotype because there are two different single nucleotide polymorphisms within the APOE gene that contribute to, there are 3 protein alleles, E2, E3, and E4. E4 increases your risk of Alzheimer's disease in a dose-dependent way. So if you have 1 copy, you're about a 3-fold increased risk. And if you have 2 copies, the estimates vary 8, 10, 15-fold increased risk of disease. Very high, apparently high increase, high risk if you have 2 copies. And then the E2 decreases your risk. So ApoE3 is actually the most common. 80% of alleles of ApoE are E3 in most human populations. E2 is about 7%. And if you have 1 copy of this, you're about half the level of risk of someone who is a 3-3 genotype. So over here you can see, this is from a review from Alan Rose's. Here's age at onset, and here's the proportion of each genotype who is still unaffected. So at 60, pretty much everybody is unaffected regardless of genotype. But if you look at the 4/4s, in their data set, by the time you reach 75, 80, almost 100% of the 4/4s were affected. So this is a very biased data set, I have to say. This is not population data. But you can see that there's clearly a difference. So here's 3/4. And if you look at the other end of the extreme, so here's 2/3. By age 80, maybe 50% of these people have developed Alzheimer's disease, whereas it's close to 100% of 4/4s. Yes.
Why do you think 2s are so rare?
In the population? Well, so APOE4 is actually the ancestral allele in the sense that if you go back and look in other primates, the sequence is most similar to APOE4, right? So in human populations, 2 and 3 have arisen in human populations, and APOE3 must have massive positive selection on it because it's now the most common allele in any human population. So it doesn't answer your question, but I mean, I think, in terms, Alzheimer's disease is never going to be the selective pressure, right? There's no, they're all getting it after they've reproduced, so there's no impact. But there is something probably related to lipid metabolism, maybe diet, that was influencing the frequency of these polymorphisms in human populations over evolution. And I think it's really actually very unclear what the driving force was at this point. And there has been some discussion in the literature about whether it's related actually to infection, and because there's some data suggesting that people with E4 alleles have different propensity to infections in tropical climates. But I would say at this point is really not clear what drove this frequency. Shay?
So what about the E1 allele?
Yeah. Yeah, I mean, basically, it's not common in any populations, and there are obviously these are the major ones. There are some versions of these other alleles as well, where there've been other mutations on them. That in the Colombian kindreds with the Presenilin 1 mutation, there are some sections of that family where they have another APOE. They have an APOE variant that's really unusual. I don't know whether it's affecting risk in those families. So the predominant effect of APOE, would say, is influencing age at onset. And you can see here that people with the highest risk alleles are more likely to have this earlier onset of disease. Yeah.
These families, do they also show the same proportion of E4 carriership as well, the prevalence, and does that have an additive component to it, or can you not even...
So that there is a literature on this. Probably the easiest thing to pick up is that people with E2 alleles in these families look like they might have a later age of onset. So it's probably having some effect.
And in our experience, the ones with E4 sometimes had an earlier age.
Yeah, I mean, and we've reported that too, but there are discrepancies in the literature around this, and I think, exactly. The sample is small, and the impact in different mutations might be, maybe more apparent. So we did it first looking in those Colombian kindreds where everybody has the same mutation, so you kind of controlled there, whereas if you look across many different families with different mutations, it's kind of harder to see. But I do think there is an APOE effect, yeah. Okay, I think we'll just... So let's try, and I'm going to skip a lot of this, but I just want to introduce this since, obviously, you're going to run a GWAS. So genome-wide association studies. So the beauty of these is that it allows you... So now we're going to go really deep, right? What we were doing in linkage was actually probably of the order of hundreds of DNA markers covering the whole genome. Now we're looking at millions simultaneously. So this was a huge breakthrough, actually, I think, for genetic studies that allowed you to be able to genotype many more variants in the genome. So you're assessing many more points in the genome. And because you can do it on an array, you can do them all simultaneously, and you can do it in thousands of people or tens of thousands of people. So that really opened up the kinds of studies that you could do beyond the family studies, right? So these genome-wide association studies, you evaluate millions of DNA variants simultaneously. And the most common design is a comparison of unrelated cases and controls. So you're really just asking the question, does this polymorphism, does the frequency of this polymorphism, is it different in the cases versus the controls? But you can obviously use a family design too, and that's what we did in the NIA load families that we collected. Or you can use endophenotypes. So quantitative traits, so in ADNI is a good example, right, where you have imaging data, you have fluid biomarker data. You can use any of those phenotypes as quantitative traits in an analysis, a GWAS in the sample. And the most important thing of GWAS is, oh my goodness, right, is you need very large sample sizes, right? And that increases power. And I think we've seen that in the Alzheimer field. The first GWAS that were done in Alzheimer's disease were like 1,000 cases and 1,000 controls. We found ApoE lots and lots of times and nothing else, right? Schizophrenia, they did 10,000 people, never found anything. Now they have 100,000 cases and controls. They have 100 loci. Sample size is everything. So big sample sizes increase your power.
Alison, how does one do a power analysis on a GWAS? I mean, is there like a rule of thumb that you use?
My power analysis as a non-statistical person, the largest sample size I can afford. There are methods, there are calculators to do it, but it's all making assumptions about the inheritance, which we don't know anything about, really. So they satisfy reviewers, but I don't know that they're necessary because it makes a lot of assumptions that may or may not be true.
We'll see more tomorrow on that.
Okay, right.
With some allusion to programs that allow you to conduct those.
Yes, right.
Deep dive by name.
Yes, yeah, right.
It's like a very
Yeah,
main question,
fair.
but is the idea with the large sample sizes due to this assumption that the effects are going to be small, or is it a problem on the case control side that there's noise?
There's probably a bit of both, I would say. I mean, I think that we clearly know that the effect sizes are small. So that's a big part of it. But I think we shouldn't discard the possibility that at least a proportion of it is due to contamination with non-AD samples in our case, right? Or, so a misdiagnosis, which could be misdiagnosis of a case because you have a different dementia, not Alzheimer's, or it could be you have controls in there that have some underlying disease that hasn't been detected for whatever reason, right? So, yeah, I think there are multiple reasons why that might be true. And so, that sort of, a quantitative trait then is valuable in that regard because you're not having to make a decision one way or another about, it's something that you can measure in everybody and put a value on. Whereas cases and controls, you're trying to force people into one of two groups. There are some people who don't really fit, and so you have to either generally exclude those people, obviously, if they were MCI and you're not sure whether it's MCI due to AD, then you probably throw those people out. But you're still going to have some contamination in your sample. There was a question over here somewhere as well. Was it the same question?
Well, I was actually going to add to that to say when we look at things like the educational attainment GWAS, for example, which doesn't have the case control contamination.
Yes.
You're definitely seeing now in the third version of this that the increases in sample sizes are definitely increasing the number of loci. So.
Right. Yes. And I mean, I think that you look at things like height and BMI and all these anthropomorphic traits that you can measure in hundreds of thousands of people. we definitely seen in those studies, the more people you add to those studies, the more loci you find.
With small effects, though, I guess.
Always with very small effects, yes. Yeah. Yeah. So ApoE is a real aberration. And it misled us genetically for a very long time, because when you do your power calculations based on ApoE effects, you need tiny sample sizes. But there's nothing like ApoE.
So we might talk about this later, but shouldn't we do a GWAS subtracting ApoE from the mix then?
So that's something that we have been doing, or doing it in different ways. And I guess, okay, let's get to a Manhattan plot. We need to at least have one of these, right? So this is, every GWAS is accompanied by one of these, right? This is a Manhattan plot, because you have some skyscrapers like ApoE, if you're lucky, right? Some of them look more like Kansas, I've seen. Right. So chromosomes along the x-axis, minus log 10 p-value on the y-axis. And then this red line is the multiple test correction for the number of SNPs. Right, so 5 times 10 to minus 8 is what is most commonly used. So any of you who work on Alzheimer's disease have probably seen this slide hundreds of times. So this is from the IGAP consortium paper in 2013. And there were, in this study, 20 loci that lie above this line. And that was based on data from 74,000 samples. So if we add more samples, some of these other signals will probably go above the line. Right now, they're below the line, and so we don't know whether they're real signals or not. The other thing I want to point out on this is that all of these are so they're labeled, which is sort of comforting. It seems like somebody's identified something very specific, which is affecting risk for disease. You should ignore that information. It's not, in some cases it might be correct, but it's definitely not in some others. These genes are the closest gene to the SNP, or single nucleotide polymorphism, with the lowest p-value. But, as you will see in the next couple of slides, sometimes these loci contain lots of genes, and somebody arbitrarily assigned a gene. And so this is incredibly misleading. I actually hate the fact that it's labeled like this. It should have been labeled with the RS number of the SNP, because that's the only real piece of information that this is showing you. So now, somebody asked me a question that was, I've forgotten what the question was. Oh, stratification, yes, right. So this was done with the whole data set. And there have now been, so there was published, I think earlier this year, or no, well, maybe last year, a GWAS that was done with the same data set, split into E4 positive and E4 negative individuals. There wasn't a huge amount of difference between the results, with the exception of the tau locus that only showed up in the E4 negative individuals.
But isn't that really, really surprising, since there's two major pathologies of Alzheimer's disease, one of which is tau, and it like hardly ever shows up on
Right. Yes.
That's my problem with this, I guess.
Well, so, I mean, if the tau pathology is a consequence of amyloid pathology, you might not affect it. I mean, so the one issue that I have that I don't think we really know the answer to in the E4 negative group is whether the tau locus shows up because that region of the genome can influence AD, or whether these people have frontotemporal dementia. Right? I mean, I think they probably don't all, but I wouldn't be at all surprised if there is a greater contamination of the E4 negative dementia group with non-AD dementias.
So this sample is people with clinically diagnosed and confirmed Alzheimer's disease only, like symptomatic Alzheimer's disease.
Yeah, yes, the MCIs are not included in these analyses. There are very few autopsy cases, only about 2,000 in this data set of 37,000 cases. Yes, found APOE. Because it's about 2 or 4,000 people. It's not very many people, that's the problem. So, this is the trade-off, right? Imprecision, in order to get thousands, tens of thousands of people, you have imprecise phenotypes. And yes. Yes.
So, I know, like, 10 to the negative 80s, even as a non-geneticist, I hear that a lot.
Right.
What would happen if you just lower the bar a little bit? We know we already have error in our phenotype measurement.
Yes.
Why not allow a little bit of error in the genetic measurement? Allow some false.
Right. Well, you can do that if you then go on and you have a replication sample, right? The challenge here is that if the bigger the discovery sample, the more likely you're going to find things. You don't really have a replication sample because you put everything in the discovery sample in the first place. And so I think I think that it is an issue that and it's one thing that we are trying to do now by combining expression data with genetic data, with lowering the threshold and then asking, can we identify which of these sub-threshold findings actually have a pattern that's similar to? So to sort of emphasize the point that you shouldn't believe the gene labeling, right? This is a good example. This is now blowing up one of those GWAS hit regions and looking at it locally. So here is the signal. And that signal is all within the SOL1 gene. There's no signal in these other genes down here. And there's no gene in this bit here. There's 2 recombination events that are pretty strong here. So that suggests that it's going to be located here. SOL1 affects trafficking of APP. It traffics a lot of things, but APP, if you knock out SOL1, you affect APP processing. So this is a pretty good candidate gene, and the signal is right over one gene. So that's kind of the best case scenario, but it is still technically possible that one of these SNPs regulates something else over here, and they've been caught out in other diseases where they have focused large amounts of effort on the gene that's right underneath it, only to find later on it's regulating something down here. So you always have to be really careful of that. And most regions look like this one. Go on.
And that is a strategy, though. So to follow up on that is that you take the p-value, you use a bigger p-value cutoff, which is what you're suggesting. Genome-wise, you cut off, and then you can take those SNPs that are and limit your number of SNPs so that you have a lower p-value, and then you don't have as big of a bond-forming correction to do. And then you take those and then try to replicate that, so you can get another replication or data set, and that kind of is another strategy to try to locate whether or not it's a true signal.
Yes,
or not.
right.
So looking at those 2 strategies,
Yeah.
either throwing your whole sample into a single large sample and doing your GWAS versus doing a development and validation cohort, have people done simulation studies to see which is the stronger design for finding value in genetic stuff in a genetic?
In the past, for sure, it was always throw it in the kitchen sink. Go with the biggest data set possible.
So a lot of people, you can't publish anything off of just one data set. You need a replication, or what you do is then you then take the 2 together and do meta-analysis afterwards, and then see where you're sitting.
Right. So, this IGAP study is a meta-analysis of many different data sets.
There's other ways you can do it, so by looking at the functional status of the SNPs and doing different p-values for one, or SNPs that fall within an exon versus ones that fall outside genetic regions. Not very common yet, but people have done that to get differential p-values within the same study.
I'm going to skip. Oops. Okay. All right. So just a couple of minutes. Just to say, so we're talking about GWAS, and that's finding common variants. Right. The other thing that's now happening is there's a lot more whole genome and whole exome sequencing. And so if you want to find low frequency or rare variants of moderate effect, then you use a sequencing strategy. And you could be targeted sequencing in the old days, but now that's just as expensive as doing a whole exome sequence. Right now, whole exome sequence is probably half the cost of doing whole genome, but obviously you're getting 1% of the genome that you get when you do whole genome. So it's the same argument that you had when you were first doing a GWAS and how expensive, how much money do I have, and what can I do? Now we're having the same discussions about sequencing, and my preference actually is really to do as many people as I can do, and I would do whole exome. I got told by my program officer, I won't sponsor you to do whole exome, you have to do whole genome. So just a word of warning to you, so you then have to compromise on your study design. So the other way that we have done things...
Sorry, what's whole exome?
Oh, sorry. Okay. So if you remember when Tim was talking, thank you, he talked about introns and exons in genes. An exome is just the exons. So it's the 1% of the genome that's coding for protein. And you can, now people designed capture reagents, so you can capture just that DNA. So you just sequence that 1% that has that. So the other thing that people did a few years ago was make an exome chip, which is a chip that has all the variants on it that had previously been reported in the genome, on a chip that looks like a GWAS chip. And so you can then just do it genotyping like that. It's limited by the fact you're not going to find anything new in terms of variants, but it's very cheap. So a chip that's got GWAS and these rare variants on costs about $50. Costs about $1,300 to do a whole genome. Yeah.
So, can you give us a sense of the amount of data that's coming out? So you've got SNPs, millions.
One whole genome is a terabyte of DNA. Right.
Well, SNPs, it's a million SNPs for a GWAS or something. So
right.
how much data comes out of the whole exome sequence? What are the things that you're analyzing?
Well, obviously, so you're getting sequence at every base in that 1% of the genome, right? But if you then process that so that you turn that into a file that is only calling the variants that are different from reference, then you have a lot fewer, you have a relatively small, a much smaller number. I mean, you've got a call at each base, in fact, but most often it's going to be reference.
Okay.
So you're basically turning exome or genome into a genotype file, like from a SNP array, but you're now calling every single base. So 3 billion bases, you're calling every single site. I mean, obviously some of it's repetitive and it's hard to call, but close to that's what you're aiming for, right? So this is data from actually earlier this summer, reporting these are now rare variants that are amino acid substitutions in these 3 genes that are rare in the population but increase risk for Alzheimer's disease by about 2-fold. And one of them is protective. I can't remember which one it is. I think it's that one. And it's decreasing your risk by about to about 1/3. So this is a cheap, easy way, but it has the limitations of only having any variant that's already been identified in the genome. If you want to look for novel variants, then you have to do sequencing. And so these are the genes in Alzheimer's disease that are now being implicated by sequencing. And TREM2 and ABCA1 have really come out of multiple studies. These both contain loss of function alleles that increase your risk for Alzheimer's disease. The other ones, SOL1, there's a bit more data on this, and that, again, and there's some loss of function variants in that. The other ones are really 1 or 2 studies at most. So there will be lots of data coming out on those. And I'm going to just skip now to... so all of this will be online if you want to go through it. I guess this... this is a summary then, right? So there are more than 20 loci from these GWAS in Alzheimer's disease. This I didn't go into, but I think it's a really important point in terms of thinking about how you go from those GWAS findings. If they're only really reporting regions of the genome, how do you figure out what's important in that region? And doing pathway analyses, it's pretty clear that these GWAS loci are enriched for genes that are involved in 3 major processes: immune response, lipid metabolism, and endocytosis. So those biological processes probably underlie risk for Alzheimer's disease. And one of the things that we notice is that among these genes that are under the GWAS loci, they're highly enriched for genes that are expressed in microglia. So those are the cells in the brain that are the immune surveillance or surveillance cells, that certainly fits with this. One thing you should think about, so we found risk and protective alleles in both the sequencing and the GWAS, and you find, especially in the sequencing, you find different variants in different populations, right? So if you sequence 1,000 people for a gene, you take 1,000 people from a different population, you won't find the same variants. You're going to find, hopefully, if the gene's real, you'll find different variants in the same gene, enriched in the cases in that population. And so we're going to really need a much more complete atlas of these risk alleles to be able to completely figure out what the networks are that are contributing to disease. Okay.
One question.
All right. Yep.
May I ask one question? A burning question. What proportion of Alzheimer's cases is familial?
Well, a little bit depends on how you define familial. But, I mean, I think if you was, I think that if you're asking for, at least one first-degree relative, then probably close to 40-50%. As you increase the number of relatives that you're requiring, of course, that number drops off dramatically. But, with ApoE having such a big effect, if ApoE is in your family, then you're probably going to show some familial aggregation if you have ApoE4 there.
Well, terrific talk.